Thanks, Dan, for that excellent perspective. So we're going to now move into the industry part of the stakeholder presentations. And I'm pleased to introduce Emmett Schmidt, who's ex from Merck and has an excellent window into the interaction between companies and the patterns of choice that are made around potential development of combinations. So, Emmett.
So, is this okay? Yeah. So I'd like to thank the organizers for inviting me to give this talk, and particularly Roger. Roger didn't note that he offered me my current position, and he offered it to me a little sheepishly. What he didn't say is that I'm offering you a job to be a professional spaghetti thrower. And having just spent the weekend with my 4-year-old granddaughter, twins, I will say there's some virtues and some aesthetics in spaghetti throwing. And just consider this talk an ode to spaghetti throwing. This is a slide which Eric Rubin has been maintaining in his own personal search of clinicaltrials.gov over the last 4.5 years. This may be the quintessential ode to spaghetti throwing. And I'll note that there's an underlying rationale behind this that's not represented in this room. I'm intrigued to note that the investment community isn't sitting here, and yet a good deal of what probably is accounting for this massive increase in trials is the massive investment that's ongoing in the investment community. My job is this. This is a waterfall plot of all the known pembrolizumab monotherapy activities across a variety of indications, and this is truly jaw-dropping. I think if you don't, if your jaw doesn't go wow when you see this, then I don't know what would make your jaw drop. But it immediately says what the problem is. The problem is, of course, all of those lines that are going up. The unmet medical need is now residing there. And there's two pretty simple principles for how you go after that. One is biomarkers, of course, and you just address those tumors which are in fact reacting, or you find something that makes the other side go downwards. If nothing else, please take my talk as the null hypothesis talk. You're going to be hearing an awful lot about immunology, and I deeply respect immunology. I think the immunologic community, having been beaten up for all but the last 5 years, probably has their turn here to really speak up for themselves. On the other hand, we've been, in fact, combining therapies for 50 years. And if you go back into textbooks in 1920 and 1940, which is what Adam Palmer did in this lovely paper in Cell, we have other principles by which you combine drugs. And it's really very simple. And it's depicted in colors, in the purple and the green, and it's called independent action, and there's mathematics for it. Just as there are mathematics for the combination index, the Chow and Tallali approach, et cetera, that systematically can be done in a cell culture model, not so well in mice, and very difficultly in humans. But having said that, it's obvious that if you have independent actors, put them together in a patient population or in cells in a tumor, you can get combination effect even if they have nothing to do with each other. And in fact, you probably can do better by orthologous mechanisms because you're less likely to coincide on the same target tumors. Now, likewise, just to think out loud with you about some of the other approaches and the immunologic approaches, and everything we do has a problem with it. What I'm going to tell you has a problem with it. But we're heavily dependent on post hoc analyses when we start doing these highly dimensioned multiple array and non-multiplicity corrected looks, retrospective looks at biomarker data. Similarly, experimental model-driven approaches to this also have, their vulnerabilities. And this is probably the easiest way to show you the vulnerability. There's really a very limited number of these mice that you can do these experiments in. There's really only 4. And those 4 are far from representative of what the human situation is across human beings, across the indications that we look at, and across the immune environment of all those human tumors. So, the idea that those 4 mouse models are going to be able to drive us quite so far forward at least needs to be thought about deeply and looked retrospectively from what I'm going to walk you through. Being deeply respectful of the history of oncology. I'll just point out that as a pediatrician, we've been curing leukemia for quite some time. And it was cured by virtue of a variety of thought processes. Of course, I'll invoke a few of the heroes of early oncology, Emil Frey, with the idea that with enough serial log kills, you could get down to 0 in theory. I suspect that was actually immunology at the baseline somewhere underlying that, but at the time they didn't know about it. And I'll point out over in the left lower hand corner that that's the current approach that we have for a combination therapy in pediatric leukemia. And I defy you to think how you could have arrived at that combination in mice. It was done in the lower right-hand corner. This is the Children's Oncology history of oncology over 30 years with overall survival, one after another of pretty straightforward, simple trials. And they all look like the upper left-hand corner. They're simple add-ons. So, what played out for Merck over the last few years, even extending slightly before Roger arrived, but could not have happened, of course, without him, is the simple add-on. So it was just take standard of care, whatever the standard of care is, and add pembrolizumab on, and that was 21G. And the lung team that wrote that trial are my good friends. It's been fun to watch them succeed against an awful lot of people saying it wouldn't work. And by gosh, it did. This is Keynote 189. I think each of the other speakers have pointed out just, how impressive this result is. And I think it's a really important result in the perspective of how you're going to interpret this. So this is a slide stolen from Roger. So what have we been doing? Within Merck, we are trying to take a data-driven approach to this. As it turns out, when Roger allowed me to become the professional spaghetti thrower, I set in play 2 principles for how we would do our collaborations. So without really knowing it, I was sort of performing a systematic, not randomized experiment. So half of the trials, as it turns out, have been motivated by combining things that had their own activity. As shown in a phase 1 trial, doesn't matter how it works. Most of those are going to be standard of care and targeted therapies. And/or, with the deepest respect for immunology and needing to parse immunology as simply as possible to be a lumper, to be able to give you this talk, let me borrow the chance to just say I'm going to divide all of immunology into 3 parts. They will be innate immunity, and although this one is confidential, this is my own hand-drawn diagram of immunology, so you can ignore that confidential part. Adaptive immunity and the tumor microenvironment, just crudely to begin to have a scoring system. I haven't been doing this idiosyncratically. We actually have a team within the collaborations team that assigns these categories. This isn't Emmett's personal opinion. So this actually has representatives of discovery, clinical, and the early phase oncologists who assign these. And interestingly, on the left-hand side of this are all of the potential collaborations we've evaluated in my team over the last 3 years, and there's a lot of them. And I'll note first, interestingly, out of these, somewhere around 350, probably 325 of them had positive mouse experiments. So just to note, it's not hard to combine anything with PD-1 in a mouse experiment. And just to note, I think if you do it enough times, it might even combine well with normal saline. And so the controls that you put into these experiments is really important, and the reproducibility of it. A one-off on a mouse experiment, okay. Now, interestingly, the results I'm going to give you on the right are what I've compiled out of 65 combinations with pembrolizumab, 34 PD-1 combinations with the other PD-1 molecules and PD-L1 molecules on the market, and 82 PD-1 monotherapy trials. And if you'll note, actually, the number of each of the different assets is roughly proportional to the number of assets of that type that has been proposed to us along the way. So there's not really any selection in the how we've chosen to represent these results. It kind of represents everything that's come into the landscape. This is the portfolio of our wonderful collaborators, and I'll have a further comment about them toward the end. And this is your first Rorschach test. So now these are waterfall plots, sort of to compare to the earlier monotherapy waterfall plots. And I would suggest that if you look till about the last 4 of them, you're going to see combination effect broadly. So my first comment is from this first Rorschach test, more than half, and in fact, an awful lot of things combine just fine with PD-1s. Now let's begin to sort of tease this out from the data standpoint. Out of all of the data that out of all that trial number, this is what I could pull out post-ASCO. And if you'll note, there are overall response rates increasing in virtually any proposed mechanism that you might consider. So the idea that there's going to be something special here is challenged. Now, this is an odd depiction because, of course, the monotherapies are over on the right. And so what you pick here as your eye to decide whether your combinations are working or not might be the median of all of the monotherapy results on the right-hand side. And in fact, that turns out to be a bit of a challenge in trying to interpret the data. So this is a list of all the monotherapy results, and the problem is obvious, which is there's 6 different ways you can get monotherapy data. 3 lines of therapy, each of which are either PD-L1 or other biomarker enriched or not. So there's a degree of subjectivity in my next slide that you'll just have to take my word for it. I assigned as best I could a monotherapy comparator based on line of therapy to each of those combination results, and this is what you get. So this is simply the combination ORR divided by the monotherapy ORR in the equivalent indication and line of therapy. And I think this begins to make your data a little bit more in a place where you can sort of understand where we are in phase 1. Roy, of course, is going to point out correctly that we haven't tested these in phase 3. Needless to say, this is a stepping point to phase 3 and arguably might be another way of looking at this that's not mice. And at least in this moment, the things that are showing promise are in every possible way you can do this. And by the way, probably roughly proportional in every way you can probably do this. So chemotherapies of many types work fine. Targeted therapies of many types work fine. Maybe the tumor microenvironment may be lagging a little bit. Certainly adaptive immunity, which is heavily dominated by the CTLA-4s, is working. And last, there's plenty of things working in the innate immune system. Interestingly, here you're probably looking at a skew toward melanoma trials. But here is my first interpretive slide. So everything in red is a combination with a drug that on its own had monotherapy activity. And I would suggest that at least from the standpoint of an initial cut of the data, that it's a good idea to combine with things that have their own monotherapy activity. So Roger, I hope I passed the first test when you allowed me to throw spaghetti, choosing to in fact engage in working on these targeted therapies to prove to be a good idea. The question here, of course, is what's in green in the epacadostat trials, which I think are going to be a really interesting phenomenon. I think it may be the most important negative result to emerge. Now here again is another sort of first cut at the independent action model. So this is a little complicated. Everything in orange in the background is a PD-1 monotherapy contribution, and this is arbitrary because the monotherapy effect of pembrolizumab in first line is known. The monotherapy effect of atezolizumab and nivolumab is known in second line. So I've arbitrarily assigned those for the lines of therapy on the left-hand side. And then the black lines are the known monotherapy contribution of the various agents there. Those are also imperfect because data exists in second line for some and first line for others, so it's imperfect. But having said that, I think that you can see that, at least for all the way over to where your last monotherapy has activity, you're seeing combination effect broadly. And where you're not actually seeing much combination effect is with those agents that had no monotherapy effect on their own. So let's talk about epacadostat. I think epacadostat was, at least I was giving this talk up until 2 days before the results were out there, and I gave myself liberty to use the word synergism. Something which has no monotherapy activity that has combination activity is probably a pretty good case for a candidate for it. And in fact, it was based, like all of these other studies, on very nice experiments, both genetically in mice and in tool compounds. And likewise, this is a fun study. This is 037. It was one of our portfolio and is ongoing. But it was one thing to look at the overall survival superimposition. This is truly breathtaking in the other way around. The idea that you can so thoroughly superimpose a combination effect on top of the monotherapy effect. And by the way, it superimposes on Keynote 06. We have reproduced that data now 3 times. So I think we know what the result is in first line of what pembrolizumab is. But this is pretty convincingly negative if you look at it. And the important thing I think to note is if you look on the first line, what changed is the phase 1 to the phase 3 overall response rate number. So, again, in interpreting what's going on with epacadostat, there's going to be multiple hypotheses, but at least one is there was a problem with the phase 1 trial, which is why Roy is going to challenge me when we get to the next conversation. So, when I've given this talk a couple of times, Roger has pinged me and sent me emails saying, Emmett, you can't have it all that way. Can't you make this work both ways? And I think that's absolutely right. I think where we're going to achieve the greatest progress from this is probably where we use both of these sets of reasoning. So yes, chemotherapy has some immune effects. We're going to need to understand those immune effects, but they at least weren't as bad as was postulated by the people who were critiquing the lung trial and the lung program at Merck. And interestingly, there are other places where our approaches are looking promising for coinciding. And at least for the moment, our poster child are VEGF receptor TKIs. This has been reproduced across multiple companies. All the companies have seen this when they've put it together. And I picked the one place where I could find waterfall plots for both lenvatinib and pembrolizumab, and the combination waterfall plot for the combination of those 2 drugs in endometrial cancer. And it's, our results are compelling. I think anybody who's at Memorial Sloan Kettering is aware of this. And have been watching this play out over the last 3 years. And in endometrial cancer, clearly an area of unmet medical need, I think this is enormously exciting. And this is also mechanism-based. So if you actually look for VEGF receptor expression, just do this really simply, across immune cells, you'll find VEGF-R receptors on all of the immune cells. And it's presumably there on an evolutionary basis to do something. And you can put immune cells through their paces by treating with VEGF and achieve all kinds of results that are immune-specific for VEGF activity. So I'll just close with 2 things. One is to point out that there's going to be several places where independent action and immunity are going to coincide, and I hope we pay careful attention to both hypotheses. That independent action is a null hypothesis to the immune has a good argument for supplementing it and working together. And the last thing I'll actually say is it's not hard to work as company to company. I left academics 7 years ago. I've had the most delightful ride of my lifetime working for Merck. And by the way, every one of these companies is a person that I'm on the phone with. And we have a lot of fun. We're having enormous fun working together for the benefit of all those patients whose unmet medical need is out there. And the virtues of doing this in a collaborative way, company to company, I think have sort of probably obvious from the outcome of the results that I presented. Thank you.